I'm gonna give until like 12:30 for people to join. All right, maybe like one more minute. You guys can hear me, right? Can you like confirm in the chat? All right, cool. All right, so just to make it clear, I know there's like at least one or two people who come to my regular sessions, but for those of you who are new, the thing about VSI sessions, which is I do bi-weekly on Fridays, is that you guys are not allowed to have your cameras on or unmute yourselves for privacy reasons. And that's because I'm going to be able to send this video out afterwards to students. So all of you are here, you guys can get the video after or whenever it's ready, basically. And that's something I can't do in my regular sessions. And that's why you're allowed to talk out loud or show your camera or whatever. So that's just a difference. But other than that, it's just a regular session. And for today, your test is on Wednesday, right? When will the video be ready? That's a good question actually, because this is my first time doing it. But hopefully, I know the test is on Wednesday for you guys, right? I want to make sure of that. Is it on Wednesday? Yeah, because I know for both of you guys it's on Wednesday. And I'm assuming there's a mix of Quaylor and Sovereign students in here, right? Is anyone here from Quaylor? Yeah. All right. Okay. So yes, I know both of you guys have it on Wednesday. So I mean, my editor has until like, I don't know, like 3-4 days to get it ready. So I will try my best to get it out before Wednesday. And yeah, that's all I can promise right now. But I think we are able to start right now. So for those of you who have already came to my sessions before, this is almost like a mixing pot of like all of my session questions before. And for those of you who never came before, you'll see real quick how I run my sessions. So let's start here with the cell cycle. So this is the first thing kind of that you have to know for exam block 2. So just tell me in the chat, what are the 4 stages of the cell cycle in order? Do you guys remember? Prophase, metaphase, anaphase, telophase. Now, can someone tell me why that may not be correct? Is that a state? Are those the stages for something else? What do you guys think? Do you think Tracy is correct, or do you think she's incorrect and you have something else you want to say? Right. So yeah. So what Tracy just said were the stages of mitosis, and that's good because mitosis is one of the steps of the cell cycle. But the other 3, they're kind of named differently. What do you guys remember? And if you guys aren't sure, just let me know so I can kind of help you through it. Yeah, so G1, S, G2, mitosis, exactly. So G1, oops. I think S1, G2, Mitosis. Cool. So give me like a brief description of what goes on in each stage. So what about G1? What happens there? Cell growth. Perfect. Yeah, something simple like that. So what about S? So I'm seeing mostly synthesis. Tracy, replication, I think that's more in mitosis, which I kind of spoiled. But yeah, so like the thing about these three first guys is that the letters stand for something. So G is growth, S is DNA synthesis. And what's G2? Again, you can just keep it simple. You guys remember what G2 is? Yeah, it's just more growth, basically. More growth. And mitosis, I kind of talked about it earlier, but what do you guys, how would you define mitosis? I think Tracy had it the first time. What do you guys think mitosis is? How would you define it? Real simple. Cell division. Yep. And replication. That's good too. All right, so it looks like we have a pretty good grasp on this. This one might be a little harder because you're gonna have to dig deeper into your memory, but there's like 2 genes / proteins involved with the regulation of the cell cycle. Do you guys remember what they're called? You guys remember? I'll give you a hint. In lecture, they were referred to as like the pink and purple molecules, although in real life, they're not pink or purple, but that's how they were referred to in lecture. Right, so it looks like we got a tumor suppressor and proto-oncogene. All right, good. So what do they do then? What does a tumor suppressor do? Oh, well, I guess you kind of answered it, but basically overproduction / overreplication of cells. So if you're suppressing tumors, connect that to like cell division. What does a tumor suppressor do? Think about what a tumor is and what the word suppression means. Yeah, so it basically stops cell replication. Yeah, so yeah, you guys got it right. And I'll give you a hint for proto-oncogenes. It's the opposite. So what is this? What do you guys think it is? These two guys are like fire and ice. So yeah, promote cell division. Very good. Any questions on this? No? All right. All right. So Tuyen says cyclin D. Let's see here. So cyclin D and CDK4 or /6, they actually, I believe, they suppress retinoblastoma, which stops, I believe retinoblastoma is a tumor suppressor, so it stops the cell cycle, right? So retinoblastoma basically would stop progression to the cell cycle and stop cancer, essentially. But remember, there's something weird about how this activates. Cyclin D does not like activate retinoblastoma. It activates the phosphorylation of retinoblastoma. And if you phosphorylate retinoblastoma, do you guys remember if that activates it or promotes it? Or sorry, turns it off? Does phosphorylation of retinoblastoma turn off or activate it? Not quite. Yeah, so it turns it off basically. So this is confusing because you see this arrow pointing to the retinoblastoma and you think, oh, cyclin D and CDK4 activate the suppression of the cell cycle. But what it's really doing is phosphorylating the retinoblastoma, which turns off, I believe. So by removing cyclin D, you're not phosphorylating retinoblastoma. So that means retinoblastoma is there to suppress the cell cycle. So cyclin D is not correct because you'll still see retinoblastoma activity. So now we're left with P53 and HMGA2. So what do you guys think between the two? So we got some people saying P53. And I know, usually I would ask you guys why, but it's like impossible to explain in chat, I'm sure. So let me walk through of retinoblastoma, right? Okay, so this one is like another hard part of the cell cycle, but it's going to be talking about intracellular pH regulation. So it was, you're going to need some like chemistry background, just a little bit, but it was observed that cancerous cells had a higher pH intracellularly, which means the inside of the cells were acidic or basic in the inside. Right, so it's going to be more basic. Now, there was a hypothesis, I didn't really touch on this in the beginning, but it was observed that cancerous cells had a higher pH. So would the overexpression or underexpression of NHE transporters, which one would cause a higher chance of cancer? Assuming that the hypothesis is correct. Overexpression, right? Does everyone else agree with that in the chat? Right, so it looks like most of us got it, but natural selection. Oh, Rihanna, you're kind of jumping the gun, and I'll get to that in a second, though. Drift and flow. Yeah, it's all good, Rihanna. And don't worry, it looks like you're prepared for the next question, though. But for the 4 mechanisms of evolution, briefly explain what they're like. So what is natural selection? What's going on in natural selection? What do you guys remember about natural selection? It's a simple definition. Anybody remember? And if you don't, just let me know so I can make sure. Traits that increase the fitness of an animal or species will be selected for. So that's good. What's a mutation? How would you guys define a mutation? Change in DNA, erroneous replication of DNA. I like that. I'll combine your 2 though, like erroneous replication in DNA. And just to make that super simple, it just means like an error in the genetic code. And that could cause a mutation, right? So what about genetic drift? How'd you guys define that? Are introduced to a population. So just think about like new alleles flowing into an already set population. I just think that's the easiest way to remember it. But you guys look like you're good on that. And now we're here where we talk about the 5 tenets of natural selection. Now, Rihanna, you already named 2 earlier, if you want to remember what they were. Or if anyone wants to step in and just say. So overproduction is 1. Variation. Does anyone remember the other 3? And if you have them with you, you can use your notes too. Like, this is not an exam. It's just, what does a ligase do? Attaches pieces together. It holds the DNA together. Very close. And I see what you're trying to get at. But think about this. So with that mind in your head, or with that thought in your head, think about, like, after you're all done attaching the complementary bases, what does the DNA look like and what do you want it to return to? Like, does DNA just wrap itself up afterwards? What do you guys think? And like Rihanna and Kaylee or Casey, you guys are really close, but I kind of want the more clear answer, if that makes sense. So what do you mean attach pieces together? And what did you mean by holding DNA together? And if you guys are not sure, just let me know. Right, so, and Rihanna, I'm looking at my slides right now and I know I said that, but what I really wanted to get at was that, like, it, again, and I said you were really close, but it reattaches the DNA strands so that it can reform the helix. So yeah, the reason I kind of pushed it is because you were like there, like that was like the simple answer. Give you guys like another minute. Five prime CAAGT. Right. Good job on that. CAAGT 3. Right now, and I won't make you do the long one just for time's sake, but I'm sure you guys got it. I mean, I don't think this is too hard, and you might get like one problem like this, although it'll probably be a lot longer, similar to the last one. So if you'd like to do it on your own, you can practice that. But I mean, there's only a few rules that you have to know to really get that down. So after that, let's talk about genome duplication. Do you guys remember what that is? And the name, I feel like the name almost defines it completely. So what do you guys remember about that? What do you guys think genome duplication is? And if you guys are not sure, just let me know. Duplicated the genes that could lead to more mutations. That's true, but what I want to say is it's literally just duplicate, and I may be cheating by using the words, but you're just duplicating the genome multiple times. That's all it is. And the mutations, it comes as a result of this, basically. Duplicating the genome multiple times. So we talked about, or so Rihanna already kind of skipped again about how it can lead to more mutations. So you can look at this both ways. Do you think genome duplication / mutations are all beneficial, harmful, or neutral? So what do you guys think? Do you think it's beneficial always, harmful always, or it kind of just depends. It depends. And why does it depend? Why do you guys think it depends? So you want to expand the definition. Do you have one for me, or are you asking me to? Okay, so duplicating the genome multiple times. So, how should I do this? It's okay, I'll say this first. It's like it's not supposed to happen. It's kind of like a mutation in its own sense, where genome duplication is like a mistake. But think about it like this. Let's say you want to print out your paper, like your essay or whatever, and it's just one page, solution. And think about what I just said to Dylan. What do you guys think? Role, basically. What does an antibody do? What do you guys remember? Fights off foreign invaders. So it's like a molecule, and molecule is a really broad way to put it but for now, that's fine. Molecule fights off foreign invaders. And this is just a fact that you just kind of had to memorize in the lecture, but are more antibodies produced in the primary or the secondary response? Secondary, right. And that's just something you just know. So I want to talk about vaccines a little bit. How does a vaccine work then? And kind of mention antibodies in that sense. And if you guys are not sure, just remind me. What do you think a vaccine does? Pathogens. Oh, sorry, I realized you said antigens, but that's not correct. But I'll get into that later. But you inject a weakened or even dead pathogen, which triggers the immune response. And Maya, it would trigger the primary response as well. Now, Rihanna, you weren't completely wrong, but antigens are kind of different from pathogens. So how would you define an antigen? What do you guys think? You had the right idea though. Antigens are, let's say, bad, if that's a hint. What do you guys think antigens are? And if you guys don't remember, just let me know. The proteins on the surface of the pathogen. Yeah, that's really good actually. So it's like a protein on a surface of a pathogen. And that is also true. They do bind to antibodies, and antibodies can bind to them. Now, can anyone tell me what antibodies binding to these pathogens via their antigens, what does that do to the pathogen? What do you guys think? How does antibodies binding? Eliminates it. That's like the broad answer, which is true. And I'll explain what the more in-depth answer is. Basically, it serves as a marker, basically. So when it attaches to the pathogen, that tells the body that this is a pathogen. And what it also does, it kind of groups it up. So like the antibodies will pull the pathogens to one place. And going back to the markers, it signals for a phagocyte which is like a white blood cell, to eat it up basically. So it doesn't directly eliminate it, but it kind of calls over the guy who can. So just remember that. And now, what creates antibodies? Because remember, antibodies is a response to the presence of antigens. They're custom made. So who makes them? B cells. Perfect. Yep, that's just one thing to remember. But kind of going into more depth into B cells, what is the primary response then? So antigen is sensed. I'll give you that. So how does the B cell react, and how does antibodies? Like, how does that all work together? Give me like a quick rundown of the process. What do you guys think? And to start, antigens are detected in the body. And then what happens? Then if you guys don't remember, just let me know. What do you guys think? Do you guys need a hint? All right, so, okay, so antigens are in the body, brought in by, so basically the pathogens have the antigens on them. So between B cells and antibodies, which one of these two senses the antigen? What do you guys think? Do you think B cells sense the antigens or antibodies sense the antigens? Which one of the two? And you can just give any guess at this point. Do you guys remember? Okay, looks like you guys may need a little more help with this one. So I'll just go through it. So what happens first in the primary response is that antigens are in the body, they're detected by the B cells, and antibodies are custom made for the antigens by the B cells. So antigens detected by B cells and antibodies are made. Now, what is the other product of the primary response? There's antibodies, but there's also something else that's kind of made or produced out of that primary response. Do you guys remember? It has to do with B cells, memory B cells, exactly. And memory B cells are integral to the secondary response. So kind of, you can answer it almost together. What does the memory B cell do? And thus, what is the secondary response? What do you guys remember? Remember, it's a lot faster, so kind of think about that. And if you guys don't remember, just let me know. All right, so it looks like you guys are struggling with this part. And for time's sake, I think I'll have to keep going. But what happens in the secondary response is the memory B cells now sense the antigens. But mind you, it has to be the same antigen that the memory B cell was created for. Yeah, so it detects the antigen and then makes more antibodies. More than the regular B cells ever could. So basically it just makes, it just basically has like, it recognizes the antigen now, so it can make more antibodies quicker. So that's the difference basically. It's like the number of antigens and response time. Now, let me ask you guys this. Can B cells evolve? in the sense of, can they become stronger or more efficient at their job over time? What do you guys think? Yes or no? Yes, but only memory B cells, right? Only memory B cells. So what that really what that's referring to basically is that when you're making memory B cells, there's going to be like a trial and error process. So for one antigen, multiple memory B cells will be produced, but only one, but like it's highly probable that one of them is the best. So therefore it'll select for that strongest memory B cell. But that's kind of talking about that. And just real quick, what is antigenic shift? Think about the flu and think about antigens. What do you guys remember? And if you guys don't remember, just let me know. Mutated virus. Yeah, I think that's a interesting way to put it, but it's not wrong. Basically, what you will see with antigenic shift is kind of in the name, but the antigens will be kind of shared between different species. For example, the antigen for the swine flu, the reason we humans could even get it is because different strains of virus come together to make a new type. So yeah, so what someone said in the chat is that different strains of a virus will mix their antigens and make a new type of flu, basically. So different strains come together to make a new strain. And with the variety of antigens that this new flu has, it's going to be very dangerous because it's going to be hard to fight all those new antigens, which is why the swine flu and even like the avian flu in birds was such a big deal to us. But moving on, if you guys remember what the fused genome model suggested, what do you guys remember about it? It had to do with archaea and bacteria and eukaryotes. So what did it suggest? What do you guys remember? The tree of life, kind of. I'll give you two options then. The fused genome model suggests either that the, how do you say, the archaea and bacteria kind of fuse together to become the eukaryotes, or does it suggest that eukaryotes kind of descended from a common ancestor of both, but it's more closely related to archaea? So what do you guys think it is? Which one? That they fuse together, or that, yeah, right, so it's in the name, but the theory is that bacteria and archaea fuse together to form the eukarya. So what two things do mitochondria need to produce ATP? What do you guys think? You're doing one thing right now, oxygen and food, right? So you need food for the fuel, and oxygen allows you to kind of carry out those processes that help you break down the food. So good job on that. Now, this one was kind of like a sleeper in my SI sessions. This is kind of like extra knowledge, but if you guys remember, how does the mitochondria support the fused genome model? And remember, this is referencing that bacteria and archaea fuse together. Do you guys remember? It's something about what mitochondria once was. That's probably the biggest hint I could give you. Mitochondria, bacteria, exactly. So, and I'm not sure if you have to know that for the test 100%. I mean, they did cover it, but mitochondria basically were once bacteria that was swallowed up by archaea and thus became like our cells, like eukaryotic cells. So why is surface area preferable, or more surface area preferable for a mitochondria? What do you guys think? If you guys remember, mitochondria has like internal membrane too, to increase the surface area. So why would it want to increase surface area as much as possible? Maximize number of reactions, perfect. Number of reactions. So the more surface area a mitochondria has, the more space it has to do reactions, because all of the reactions happen on the plasma membrane, exactly, as Samantha said. So this one might be kind of hard, but DNA is wound around what protein? There's a protein that is wound around when you're packing DNA. Do you guys remember histones, right? And there's multiple histones, but I just think you have to know that this is the protein that's related with it. And sorry for going kind of fast, just that we're kind of behind schedule, but yeah, so if you have to leave, yeah, just let me know and you're good to go. I'll try to finish this out as fast as I can. And remember, just email me for the slides if you want it and the video later on. But for time's sake, all amino acids have two similar things to each other. What are they? All right, amino group and carboxyl group. Perfect. So where do they differ then? There's one more group that they differ in. Yeah, so the R chain, or R group, I'll say. So what are the three subgroups of amino acids? And for those of you who are at my SF session the other day, this should be pretty fresh. What do you guys remember? It has to do with water and pH. That's a pretty big hint. Oh, the R group, for example, is like, well, Dylan, actually, I'll email you afterwards. I'll give you a diagram, all right, just for time's sake. But I'll explain that for you. So yeah, so the three groups are acidic. And acidic basic is kind of two groups, but I just group it up as one. Yeah, sure, Dylan. And then you have hydrophilic, hydrophobic. Cool. Do you guys remember what sort of atoms you would expect to find in the R groups of hydrophobic bases? So what about hydrophobics? Do you guys remember? And I'll remind you of the options you have. You either have carbon and hydrogen or NOPS, which stands for nitrogen, oxygen, phosphorus, and sulfur. So what do you guys remember about hydrophobic groups? NOPS is in hydrophobic. Actually, I think it's carbon and hydrogen. And NOPS is the other two, actually. Although I'll fact check that later. But this one will be kind of hard too, but do you guys remember what kind of bonds they can make, respectively? You either have no bonds, ionic, and covalent, I believe. Sorry, ionic, hydrogen, and no bonds. So can hydrophobic make any bonds? Carbon and hydrogen. No bonds, right? And for time's sake, I'll have to kind of run through this, and I apologize for that. I just have a meeting after, but hydrophobic groups cannot make any bonds. Hydrophilic groups can make hydrogen bonds. And the last group, acid-base, they can make ionic. And there's two modulators that can change the shape and thus the function of a protein. What are the two discussed in class? What do you guys remember? Starts with the A and one other starts with the C, I believe. Covalent allosteric. And does anyone remember what allosteric does? Does it promote or inhibit protein activity? Think about like the hemoglobin example, if you remember, of how when allosterically modulated by oxygen, the hemoglobin started carrying more oxygen. So does that sound like promotion or inhibition? What do you guys think? Are you guys not sure? Well, I'll tell you right now, allosteric, I believe, promotes protein activity, while covalent, an example is phosphorylation. It's more stronger, basically. So it can regulate it as opposed to just promoting it. So it can inhibit or promote. So regulate, inhibit, promote. Right. Actually, that's all I have for you guys today. I was going to do a Kahoot, but we're out of time. Yeah, so email me if you have any questions. Dylan, email me if I know you had a question earlier. And yeah, for the slides, just email me for anything. But that's all I have for you guys. Sorry for taking too long maybe, but if you have any questions, please let me know. And thanks for coming.